Welcome to this webinar on biocompatibility of drug delivery devices. In this webinar, I will give an overview on how to approach safety evaluation for medical devices. My name is Lise Lyck. I work in DHI Environment and Toxicology as a senior toxicologist. DHI is an independent, private and non-for-profit organization founded in Denmark, and we work as consultants helping companies overcome regulatory barriers on a global scale. Our business is research and expert consultancy, and we help companies and authorities. And in our department, we work with regulatory toxicology and product safety, including biocompatibility for medical devices. But we also help companies ensure compliance for industrial chemicals, biocides, medical devices, food and food contact materials. And we develop software tools for chemical compliance and prediction of toxicology. And then we do all kinds of risk and safety assessments for chemicals and impurities and migrating substances for medical devices and for instance, pharmaceutical packaging materials. The agenda for today's webinar is that firstly, I'll give you an overview of the requirements for safety evaluation according to the European Medical Device Regulation and the ISO standards on the area. Then I'll move on and look more into the requirements in the ISO 10993 series for biological evaluation. Last year, an updated version of ISO 10993 Part 1 describing the overall framework for biological evaluation of medical devices was issued, and there are some hot topics or basic topics regarding categorization of devices, how to assess toxicological endpoints in the risk assessment, how to perform the physical and chemical characterization and non-clinical tests for biocompatibility that I would like to share with you. Then I'll move on into a case to exemplify it all on how to do biocompatibility and make the strategy for drug delivery device, including how to do the categorization, which endpoints to consider, and how to select the tests. The overview of requirements to safety and biocompatibility evaluation for drug-device combination products. When you consider a drug delivery device, there's a spectrum of product types which all have differences in respect to which biological risks they pose to the body and to the user. And it depends on the way that the device functions and the way that the device comes in contact with the body. But overall, you could say that a combination product is a product that has either a drug or a biological active substance, which is delivered to the body by use of a device, a medical device. And it's the device component that we're going to discuss today and the safety of that. The safety of the drug and the biological active substance is covered by the evaluation of toxicity for the active pharmaceutical ingredient or for the biological itself. So that's a different topic. But now we're talking about the device biological properties. And it differs whether it's an inhalation device like these inhalators up here, a device for delivering a drug to the nasal cavity, or injection pen. But it can also be a simple device like this pen that's used to deliver a vaginal pill, or implantation devices like this this hormone rod for implantation, or an implant device for delivery of a medicinal substance. So all in all, these devices have a very wide range of potential biological risks inherent to them and need to be characterized in a different manner and evaluated in a different manner, which is still systematic. And the regulatory framework for assessing and documenting the safety of a medical device is the medical device regulations. Currently, it's the Medical Device Directive in the EU that is the valid regulation. But from May 2020, the Medical Device Regulation will be enforced and be the regulation to consider when you have a medical device. And then for both the Medical Device Directive and the Medical Device Regulation, there's a series of guidelines and harmonized standards that are issued by ISO and by the European standardization committees that cover how to document and evaluate the safety and quality aspects of medical devices. That is the ISO 13485 on the quality management systems for medical devices, the ISO 14971 on risk management for medical devices, the ISO 14155 for clinical studies and how to apply good clinical practice when studying clinical effect of medical devices, and then ISO 10993 to assess how to assess biological evaluation for medical devices. And then there's a number of other ISO standards that are harmonized against the Medical Device Directive. Currently, no standards have been harmonized for the Medical Device Directive, but it is expected that European standards will be harmonized also for the Medical Device Regulation. So in the Medical Device Regulation, one of the essential requirements on general safety and performance is that the device shall be safe and effective and shall not compromise the clinical condition or the safety of the patients or the safety or health of users, or where applicable, other persons. And that's where biological evaluation comes in, because this is one of the means to avoid that your device causes unwanted biological effects or side effects by the use of the device. And overall, you could say that in the Medical Device Regulation, safety and performance of the medical device is the key that you need to document when you want to launch a medical device on the market and have it CE marked. So moving further on into the Medical Device Regulation requirements, in Annex 1, Chapter 2, there is a number of points which needs to be fulfilled when considering the safety and documenting the safety of a medical device. And here I have provided a short summary of those different points. So it's not the full text, but rather a summary. And one of the first things is that in bullet 10.1, there's a listing of a lot of physical and chemical properties that can influence the biocompatibility, toxicity of physical compatibility of the device, and that you need to consider and document if the device is suitable for the application and safe for the application where you use it. So that's an important bullet to know. And then there's in bullet 10.3, paragraph 10.2, there's requirements to minimize the risk posed by contaminants or residues from the design, manufacturing, or packaging of the device, meaning that you have to consider how the manufacturing of the device occurs and if any residuals can influence the safety of the device. That also includes residues from sterilization, for instance. In section 10.3, the requirements for safe use and compatibility with other materials and substances or medicinal products is listed. And here, obviously, the compatibility with the medicinal products where you use the device is the key that you need to document, the key point that you need to test and document, that your device is suitable for delivering the kind of medicinal product that you are intending to use it for. In section 10.4, there is an updated requirement compared to the MDD. Because in this section, there are requirements to reduce the risks caused by substances or particles, including wear debris, degradation products, and processing residues that may be released from the device and into the user. And when looking at the requirements, it's especially those substances that have carcinogenic, mutagenic, or reprotoxic effects or endocrine disrupting properties that are in focus, as well as particles. And it's only certain types of devices that are covered. So the device needs to be invasive and in contact with the body, or delivering uh, the body fluids or gases or medicinal substances or blood products to the body. And that's where drug delivery devices are actually covered by this point. And you need to consider if your device could contain substances that are carcinogenic, mutagenic, or reprotoxic, even residuals in concentrations more than 0.1% of the weight in a component or in the device itself. If so, you need to provide a justification and label your device with to inform about the presence of these substances. So that's quite a new and critical new requirement in the medical device regulation. In the last two sections in Annex 1, Chapter 2, there's requirements regarding reducing the risk of ingress of substances into the device. This relates also back to the packaging and ensuring that it does not become contaminated. And then lastly, in section 10.6, there is requirements to reduce the risks related to size and properties of particles that can be released by the device and into the body. And that includes actually nanomaterials. So if you have a device that is coated using a nanomaterial, you need to consider if you have to do tests or somehow document that nanomaterials are not released to the body. In the ISO 10993 series, that is the series of ISO standards and European norms on how to document biocompatibility and safety of a medical device. And the overall standard is ISO 10993 Part 1 that provides the framework for how to do the biological evaluation. But there's also a series of substandards in the series ranging and covering topics such as animal welfare requirements, how to test for genotoxicity, carcinogenicity, and reprotoxicity, tests for interactions with blood, which is quite important if you have devices that contacts the blood path, tests for in vitro cytotoxicity, tests for local effects after implantation. So if your device is implanted, you need to consider that standard. Residuals after ethylene oxide sterilization. If you have a degradable product, then there are also standards for this, and a standard for tests regarding irritation and sensitization, systemic toxicity effects, how to prepare your samples when you want to do the biocompatibility testing and toxicological testing of a device, and then I'll skip to the last two standards in the table, namely the standard on how to establish the allowable limits for substances in your device which can leach into the body, and the standard on how to perform chemical characterization of materials that is present in the device. And the latter two here, Part 17 and 18, are under active update. A new version of ISO 10993 Part 18 has been approved as a final draft ISO standard, so it's approved now and is subject to publication, expected to be published either in the end of this year or in the beginning of 2020. For ISO 10993, the standard on how to establish the allowable limits, that is a standard that's under a significant update, expected to undergo significant updates, and is in draft versions right now. So there are changes to these standards going on that I encourage you to look for. So last year, a new version of ISO 10993.1 was published, and most of you may even be familiar with it. But what was new in this standard was that there were new information on how to perform the categorization and what that means to the level of documentation that you need to provide to document biocompatibility. So especially for devices with intact skin contact, it's more specific that risk assessment can be done by comparing to other types of products and materials and other types of products for devices that are only in intact skin contact. And then a new duration category of transitory use was included in the standard, covering, for instance, injection needles that are only in contact with the body for a short duration of time, like less than 1 minute. So that's quite important as well for if you're assessing drug delivery devices that, like for instance, injection devices. Then it makes physical and chemical information mandatory, and the requirements are now more explicit. So that puts more requirements and for information on you as a manufacturer of a medical device. There are more endpoints recommended for evaluation of the device for biocompatibility, and that is aligned now with the requirements from the US FDA guidelines. So that should make it more simple to determine which endpoints to consider. There's guidance on how to perform the evaluation in a risk-based approach, and the endpoints for evaluation do not necessarily mean to be documented by testing. And that's an important point in this standard. It was the same point made in the previous version, but now in the current version of the standard, it is more explicit. It makes biological evaluation plan mandatory, and that gives an influence on the documentation structure if you already have existing biological evaluation. And then it places requirements on how to evaluate nanomaterials and absorbable materials. So that's the highlights of the new things in the standard. Then there are other standards that's new and have come this year or in the previous years. A standard ISO technical standard 21726 came out in February this year, describing how to apply the threshold of toxicological concern when assessing biocompatibility for medical devices. It harmonizes the requirements with the ICH guidelines, and I think that's a benefit. Then there's the ISO 18562 series on biocompatibility assessment for breathing gas pathways in healthcare applications. So if you are working with devices for ventilation or delivery of anesthesia gas, for instance, this is the series of ISO standards that you want to consider when you do the biocompatibility evaluation of your device. Then there are two important standards that are related to biocompatibility that are under revision and being updated. The one is ISO 14971 on the application of risk management to medical devices. That is now approved as an FDIS and will be published by ISO these days. I had news that it's now available as ISO standard. Let's hope that it will be harmonized against the MDR. And then the standard on clinical investigation for medical devices, ISO 14155, is being updated and is in this format right now, hoping for an FDIS in 2020. So important standards are under update in connection to also the MDR updates. So what's it all about, this biological evaluation? What ISO 10993 Part 1 does is that it essentially gives you a structured approach on how to assess the biological risks related to your device. It does so by giving you methods of the hazard characterization, including categorization of the device. That helps you to identify the potential biological hazards and risks. It describes how to provide physical and chemical composition of the device and can use that in your risk assessment, and it gives an outline on the approach to risk assessment. So the systematic approach is in the standard described in this flowchart, and I'm not going to go through all this very busy slide with the flowchart, but what I want is to illustrate that you can go through this flowchart, determine what is the patient contact, describe the physical chemical information and material characterization for the device, Assess the materials in relation to, are they present in marketed devices? If so, are they manufactured and sterilized in the same way? What about the physical properties of the device? Is that similar to an already marketed device? Does it have the same contact and clinical use? What does the data say? Do we have data that allows us to assess the biological risks? Are there toxicological data to assess device constituents? Does it apply to mixtures? Are the data relevant? And then you can evaluate the potential risks, decide which biological endpoints should I consider. Do I need to test, or can I justify omitting to test? Perform the risk assessment based on the sum of data that you have, and then document it all in your biological evaluation. So it's really to go through this flow chart, and then you have a structured approach for evaluating your medical device. And when you do that and consider that for the variety of devices, then you will see that for an injection pen like this one, then you have skin contact. Simply you hold the device by hand, and then you have an external communicating device, because it's also an injection pen that delivers the medicine to inside the body. Here, for the inhalation devices, they have contact to mucosal membrane, either to the nasal membranes or to the mouth, and it's also subject to inhalation of the drug that it delivers. The vaginal rod here delivers and it is in contact with the vagina mucosa, and the implantable devices here are obviously in contact with the tissues where they are implanted, and may also be in contact with the bloodstream, either directly or indirectly through the delivery of the drug. So and as you may see here, there's an increasing biocompatibility risk from the less invasive devices to the more invasive devices, and then you need to consider the risk more carefully. So you can start off by having the categorization of the devices, ranging from non-contacting devices, devices only in contact with the surfaces, so skin, mucosal membranes, or maybe breached or compromised surfaces like wounds. They can be externally communicating devices that are in contact with the blood path indirectly through injection, for instance. They can be communicating with tissue, bone, or dentin, or they can be directly in contact with the circulating blood, for instance, like a Venflon or a device that directly accesses the circulating blood path. And then you have the implantable devices that are either implanted into tissues or bones, or maybe directly implanted into the circulatory system and the blood path. And then the duration of contact needs to be considered from transitory, so that's transient contact very shortly for less than 1 minute. Limited exposure duration, that's devices used for less than 24 hours in total. Prolonged exposure devices are used for more than a day but less than a month, and long-term exposure devices are left in place or used repeatedly over a period of more than 1 month. So, and then you need to consider the cumulative use of your device. And remember that the more invasive your device and the longer the duration of contact, the higher is the potential risk of harm. And when you have categorized your device, there's a table in Annex A of the standard that illustrates and outlines which endpoints to evaluate. And that's what E stands for in this table. E stands for evaluate, not necessarily testing, but evaluating. And the only X's, the only mandatory part, is the physical and chemical information. If you have worked with the previous version of the standard, you will notice that over here in this area are the new endpoints, that updated endpoints to consider, that are aligned with the FDA guidance from 2016. So for some device types, there are more endpoints to consider according to the updated standard. And the same goes for the implantation devices. When you have categorized your device, then you move into categorizing your risk and assessing your risk. And you do so by chemical characterization, evaluation of the toxicological properties of constituents and substances that you're exposed to by use of the device. For that, you need to do the hazard assessment of the chemicals and an exposure assessment, and then perform the risk versus safety assessment by comparing the risk the risk of exposure to the hazard that the chemical gives to the body, the potential toxicological hazard, and that gives you the risk. And then you need to consider, are there non-clinical biological tests that I need to do, either by in vitro methods or in vivo methods. And the first step is obviously to do the chemical characterization. And overall, for drug delivery devices, you are quite used to that because you are quite used to doing the extractables and leachable studies for pharmaceutical packaging materials for the medicinal product. So the drug container, the vial, is almost always subject to extractable and leachable studies. But now you also need to consider, could there be extractable and leachable substances also from the device components? And you may also have to list and describe the other device parts as to chemical composition and configuration. And those chemical informations are quite important when you want to assess the biological safety of your device. So if you look at a device like this one over here, then I have made sort of an exploded view of one of the inhalation devices. And here you can see the actual medicinal substance packaging. That's the vial. That includes a pump. And then you have some external device parts here. And for those parts, it may be sufficient to collect the compositional data and supplier information to describe those polymer material components here. While for the component here, the actual drug vial, you may want to also do the extractable profile or the leachable profile. And those chemical data should be used as input for the toxicological evaluation. And now you do toxicology on materials, not just a single substance or a few substances, but maybe on a polymer material that is a mixture. And you need to consider that toxicology of materials is different from toxicology on the medicinal product. But you still need to consider the route of exposure, the duration of exposure, and the exposure dose, and that the toxicological effects can be both local, systemic, it can be allergic effects or genotoxic effects. And you need to also consider, if you have a biodegradable device, if the degradation products could, for instance, cause effects. So that's important to remember. And how to do that? There's a number of standards to work with when you do toxicology for drug delivery devices. There's ISO 10993 Part 18 that describes how to establish the allowable limits for leachable substances and substances in the medical device. There is the standard ISO 21726 on the application of threshold of toxicological concern when you assess medical device constituents or leachables. There's the ISO 18562 series that describes how to test for migrating substances that can enter from the device into the liquid or the gas when you have an inhalation device. And then there are, of course, the ICH guidelines on residual solvents, elemental impurities, and DNA reactive or mutagenic impurities, because when you assess the extractables and leachables substances from your device, which can enter the medicinal substance, then you need to consider them as impurities in your medicinal substance. Then there is the guideline on plastic immediate packaging materials, and that's for the that can be quite helpful. And of course, pharmacopoeial requirements and analyzers can be quite helpful as well in the assessment. And then when you have collected all this information, then you assess your biological risks. You consider your physical chemical properties, the toxicological data. Do I have existing non-clinical test data? Do I have clinical data or experience? If not, and you cannot account for the various endpoints, you may have to perform additional biological testing. And you should note that some authorities require some specific biological tests for devices. So for instance, the US FDA for the sensitization tests has a preference for the guinea pig maximization tests, whereas in Europe, it's quite acceptable to use the murine lymph node assay. And now I'm moving my picture again here. Then you should evaluate your overall biological risks as part of the risk management process. So you need to consider, based on our test results, are the biological risks related to use of the device acceptable? Are there any residual risks, and do we need to do additional risk management? And here there's a link to the activities under ISO 14791. So you need to document your risk assessment, and you need to keep it maintained in connection to maintenance of the product documentation. And when you, for instance, have updates to product design of the device, manufacturing products, and manufacturing of the device, then you need to also update the biological evaluation. So here comes the case to exemplify how you can make a testing strategy for a drug delivery device. So the question you need to ask yourself when you look at your drug delivery device is it actually required to perform a biological evaluation according to ISO 10993 Part 1? The basic part is it a medical device? Is it in contact with the body, either directly or indirectly? If you answer yes to these questions, then you need to consider which parts of the device are in scope for the biological evaluation, because it may be that not all parts are really in scope for evaluation under ISO 10993 Part 1, and it may be that some parts are in scope for one categorization according to the standard, and some parts are subject to categorization in another group. So you may need to assess the parts differently. And that's where you do your device categorization in accordance with the guidance in the standard. And it's important to underscore that the categorization of the device can actually be several combined by several of the categorizations. So you can have a device that, for instance, is both an externally communicating medical device because it delivers a drug by injection to the body, but it's also in contact with intact skin because you hold the outside of the device in your hands while you do the injection. Then you need to consider which biological endpoints should be addressed when you consider the biocompatibility evaluation. And then that's where table A in Annex A of the standard is quite helpful. What are the biocompatibility risks? In addition to considering the endpoints that are recommended by the standard, you may have to consider also the risk analysis that you did for the product. So that if during the product risk analysis you identify biological risks, you may have to consider them in the biological evaluation as well. And then you have to consider, how will I address these biological endpoints? How will I evaluate these biological risks? Which kind of data will I use? Will I use literature data? Will I use chemical characterization? Will I use toxicological approach, or will I use biological testing, either by in vitro methods or by animal studies? And the tests must be conducted in the final finished device or in representative parts of the device. That includes sterilization of those components that are used for the device, or if you have take only a sample of the device, or a material from the device, then you should make sure and describe how that is valid compared to the device that you actually place on the market. And in some circumstances, it may be worthwhile considering if you need to test both the drug and the device as a combined product, or only the device part in itself. So that could be that, like in the implantable hormonal rod, then you need to consider the rod material in the conformation of the rod itself, as well as the rod as it appears with the hormone inside it, because the reactions to those to the materials and to the rod may differ depending on whether the hormone is there or not. So it's quite important that you carefully consider how you want to test your device when you plan the testing. For the example, I have chosen to use once again this inhalation device that consists of the drug vial and container, the closure with the mechanical pump. It has a shell, outer shell, and a handle that helps the mechanical pump function, and then there's a cap. And obviously, these components come in different contact with the body. So those that are marked with green, the cone with the mechanical pump and the closure and mechanical pump component here, they come in contact with the nasal mucosa. That's also the parts that come into contact with the drug itself during pumping the drug into the nose. Then there are some components that are only in contact with the hand during handling of the device. That's the outer shell and the mechanical handle and pump and the cap. They are only handled by hand and may be evaluated as quite non-invasive and pose a very low biological risks. And then you need to consider how will your users use this device. That will be used maybe daily, and the users will use it either for a short period of time during active treatment, or for a longer period of time, because this is a medicinal product that is used to treat allergies, for instance. So even when considering the accumulated use, then the surface parts that are in contact with only the skin and mucosal membrane, it sums up to less than 24 hours. It's quite short term. And for the drug container and vial, then you do the extractable and leachable study to document the stability of the drug, and that you don't have migrating substances entering the drug from the vial. So that's what you do as part of the primary packaging evaluation for the pharmaceutical down here. So according to Annex A, then you need to consider cytotoxicity, irritation, and sensitization by skin contact and by mucosal membrane contact. And you can do that by having a strategy that covers, okay, I do physical and chemical characterization and description of the components of my device. So I describe what is the polymer materials in the cap, the outer shell components, the handle and mechanical pump, and actually even the label, as that is part of the outer shell of the device. Then you describe for the cone and the mechanical pump components, and especially the closure and mechanical pump component over here may consist of smaller bits that you need to describe with respect to composition of the polymer materials and the manufacture of those polymer materials as well. Then you use the material information to say, okay, is that used in a similar device that's already on the market and considered safe? If not, and if we assess, okay, we need to look at migrating substances, then you do the extractable and leachable studies. Maybe you even do that for the closure and mechanical pump component, because it's part of your documentation as a primary packaging material for the pharmaceutical. Then you use that data. You may perform cytotoxicity testing on those components that are in contact with the nasal membrane. So that's the cone and the closure. And then you assess the results. So are there any signs that these components release substances that are cytotoxic? If not, that may give a good indication that that these polymer materials are quite stable and do not give off substances. And then they will not be irritating, then they will not be sensitizing. Then you may be able to omit and justify that you omit irritation test and sensitization test. If, however, you find that they are cytotoxic or release some substances that you say, I'm not quite certain, then you may have to do an irritation test, testing, for instance, in a mucosal membrane irritation test in an animal study, or you have to do a sensitization test using an extract from the device and doing a guinea pig maximization test so as to address sensitization of your device. And then you sum up in the end, when you have the results of the studies, you sum up all of the results and assess your biological risk and report that into your product risk assessment flow. So the take-home messages from this webinar is that for doing a biological evaluation, that's the non-clinical safety package for the device component of your drug-device combination product. It's part of the technical file for the medical device component of your device, and it's required for MDR compliance, and you will also need to submit that to your notified body and have sort of that certificate when you submit your file for your combination product to authorities. ISO 10993 Part 1 from 2018 has not been harmonized yet, not against the MDD and not against the MDR, but it's still considered state of the art. It's recognized by FDA and it's recognized by a number of other authorities around the world. So I would recommend and consider using it, even if it's not harmonized. For biological evaluation, you should remember that it's different from the typical non-clinical testing of drugs and extractable and leachables for medicinal packaging materials. So there may be some endpoints that you cannot address by the non-clinical testing on the drug. For instance, implantation testing for a device component cannot necessarily be addressed by the classical non-clinical testing and toxicology performed for the drug itself. And then you have to include in your evaluation physical chemical information for the materials in the device, that you conduct a literature study and consider other data on the materials and on the device, evaluate data gaps. You should make a biological evaluation plan when you have summed up the available information and then perform your testing strategy. When you do your testing strategy, you may also want to consider, is there something on the compatibility between the drug and the biological material and the materials in your device? And you should sum up all the data and test results in the final biological evaluation report. I will say thank you for your attention. I hope this webinar was helpful to you, and I hope to hear from you. So thank you very much for your attention. Bye-bye.